Okay guys, I'm going to go over nucleic acids here. So these are the building blocks for our genetic material. So we're going to be talking about DNA and RNA. Most of the time when we talk about genetic material and nucleic acids, we're talking about DNA, which is a double-stranded ladder. Each side, I'm going to draw here in red the right side. So this right side of the ladder is going to be like one set of letters, and the letters are A, T, C, and G as possibilities. And then that will continue on for as long as up to like 3 billion letters when we're talking about the genome for people. Now, those are going to pair up on the other side with their complement letter. So A will always pair with T. This T will always pair with an A. C pairs with G. So if G is here, C will be here. This will be a T. This will be an A. So T and A always pair up together. And C and G always pair together. So if a G is on this side, C is going to be on this side. A is over here, T will be over here. So that's the base pair, base pair rule is A's and T's and C's with G's. So the difference here is when we're talking about RNA, so RNA, first of all, in the name, R stands for ribonucleic acid, and DNA over here stands for deoxyribonucleic acid. So deoxyribo and ribonucleic acid is RNA. Not too big of a difference in those two. You notice that there's a deoxy part. That means there's basically one less oxygen in a DNA structure than there is in the RNA structure. That part doesn't really matter to us too much for this course, really. Although it does change a little bit of the chemistry. So like the backbone of RNA it's a single strand, so that means it's going to be sort of open ladder. So it's like if we took the ladder and cut it in half. We're still going to have A's, C's, and G's here, but instead of a T, we're going to have a U. This stands for uracil, so it's a special base. So there's no T's anywhere in this strand. It's going to be A's, C's, G's, and U's, and it's a single strand. So that's one of the big differences between the two. One thing when we talk about DNA is that one side of this ladder is upright and the other side of the ladder is upside down. So we call that anti-parallel when we're talking about that characteristic. So what that means is DNA here can be built in this direction, and DNA here is built in this direction, downward. So it makes copying our DNA a little bit funky, but it really doesn't cause any other issues aside from that. And there's a little bit of chemistry and like structure and stuff that goes into that, but we don't need to get too much into that part as well. So this also plays into the central dogma, of course, of biology, which I've talked about before, and that is that DNA gets converted into RNA, and that makes proteins. And these are the things that actually do the jobs. So these are both genetic pieces, genetic material, genetic things that have like the code, as we would call it, or the instructions. But it's proteins that actually do the job in our cells, actually make the functions work. So I think we'll end there for that. That's good enough introduction to nucleic acids, so we'll cover the rest in class.